One of the hot buzzwords these days is personalized medicine, the idea that medical diagnostics and treatments will be specific to the individual rather than to some average patient. In particular, it captures the notion that treatments will be tailored to one's genotype, and that genetic diagnostics will be used to ascertain that genotype with respect to some disease. Today I'm going to talk about the BRCA mutations and breast cancer. The hero of this story is the geneticist Mary Claire King, who has already won the Lasker Prize, which is a good predictor of the Nobel Prize. Her groundbreaking study was on early onset familial breast cancer, which is breast cancer that runs in the family and appears at a young age. Professor King was interested in identifying the genes that might contribute to this complex trait. So what did she do? Yes, our favorite, linkage analysis. Using linkage analysis, she localized a region of the genome, chromosome 17, region Q21, that co-segregated with the phenotype in a number of pedigrees. This linkage was statistically significant with a LOD score of 5.98. It turned out that this region contained the BRCA1 gene, which was eventually cloned by a group at the company Myriad Genetics. A functionally related gene, BRCA2, that was also linked to familial breast cancer, was eventually cloned from chromosome 13. BRCA1 and BRCA2 are proteins that repair DNA damage, and thus they help to prevent DNA deletions and insertions that can promote cancer. They are considered to be tumor suppressors. Loss of function mutations in either gene that disables the protein function results in a higher incidence of cancer, and in particular, breast cancer and ovarian cancer. Severe alleles of BRCA1 can give rise to an increased lifetime risk of breast cancer of over 90%, compared to the 12% lifetime risk of a woman without a BRCA mutation. Numerous BRCA1 and BRCA2 mutant alleles have been identified, sequenced, and collected in a database over the years. Here's the website at the University of Utah for a BRCA1 mutant allele database. As you can see, there are over 1,000 mutations that have been recorded, including insertions, deletions, missense point mutations, nonsense mutations, and splice site mutations. The majority of these mutations confer heightened risk for breast and ovarian cancer. Despite the large number of different mutant alleles, BRCA mutations are relatively rare in the population. Only about 1 in 1,000 women have a mutation in BRCA1 or BRCA2. The prevalence is higher in certain ethnic groups, such as Ashkenazi Jews, who have a BRCA mutation frequency of 2%. Because they are rare, only a small percentage of breast cancer patients, about 2.5% or 75,000 women in the U.S., have a BRCA mutation. It is likely, however, that patients with BRCA mutations will have a more deadly breast or ovarian cancer. As I mentioned earlier, a biotechnology company called Myriad Genetics was responsible for actually cloning BRCA1 after Professor King had localized it to a small region in chromosome 17. This company was founded in 1991, shortly after Professor King's discovery. In 1994, they cloned and sequenced BRCA1. Shortly thereafter, they patented the gene. In 1995, they cloned and sequenced and patented BRCA2. Then in 1996, they released their first commercial product, which was used to genotype people at the BRCA1 and BRCA2 loci for mutations that conferred risk for breast and ovarian cancer. The second part of this story concerns the actor Angelina Jolie. Breast and ovarian cancer ran in her family. Her mother had both breast and ovarian cancer and died at the age of 56. Her mother's sister, her aunt, died of breast cancer at 61. Her mother's mother, her grandmother, died of ovarian cancer at 45. She used the Myriad Genetics diagnostic kit and found that she had a severe mutant allele of BRCA1, which was most certainly inherited from her mother. It conferred an 87% lifetime risk of breast cancer and a 50% risk of ovarian cancer. This Myriad test cost approximately $3,000, which was paid for by her medical insurance. Then almost exactly 2 years ago, Ms. Jolie wrote an op-ed piece in the New York Times describing her decision to have a preventive double mastectomy. By removing her breast tissue, she greatly reduced her risk of breast cancer to less than 5% from 87%. She ended her piece with the following statement: "I choose not to keep my story private because there are many women who do not know they might be living under the shadow of cancer. It is my hope that they too will be able to get tested, and that if they have a high risk, they too will know that they have strong options. Life comes with many challenges. The ones that should not scare us are the ones we can take on and take control of." True to her word about directly taking on challenges, Ms. Jolie 2 years later, about 2 months ago, announced in another New York Times op-ed piece that she had her ovaries and fallopian tubes removed to reduce the risk of ovarian cancer. She took a little time making this decision, and perhaps the desire to have more children factored in the delay. As she relates in the article, a scare concerning an elevated ovarian cancer blood marker prompted her decision. In the intervening time between these operations, an important Supreme Court ruling changed the landscape of BRCA genetic testing. As I mentioned earlier, the biotechnology company Myriad Genetics had patented the BRCA1 and 2 genes along with a diagnostic test for mutant alleles. As a result, they had exclusive rights to such a test and could charge a very high price, $3,000 to $4,000, despite the fact that the real cost of the test was probably less than $10. The exorbitant price markup led to a medical group, the Association of Molecular Pathology, to sue Myriad on the grounds that human genes could not be patented because they belong to all of us. In July of last year, the Supreme Court ruled against Myriad, stating that Myriad did not create anything. They found an important and useful gene, but that the diagnostic was just reproducing what was already in the person's DNA. Myriad tried to assert and counter-argue that the Supreme Court only invalidated their composition of matter claim on the gene, but not their method claim on the diagnostic. However, this assertion is debatable since PCRing and sequencing a gene is not a novel invention. At the end of the day, this discussion is moot because Myriad's patent expired after 20 years in 2014, and the Supreme Court ruling makes it very difficult for them to try to extend their patent. With this new window of opportunity, a number of companies have entered the breast cancer diagnostic field. Perhaps the most intriguing new entrant is a startup called Color Genomics that is offering to test for mutant alleles in BRCA1 and BRCA2 and 17 other cancer susceptibility genes for a total cost of $249, which is tenfold less than the Myriad price. This creates a new wrinkle because previously the high cost meant that you needed medical insurance to pay for the test, and the medical insurance companies would only pay for it if cancer ran in your family. At $249, you could pay for the new Color Genomics test out of pocket, whether or not you have approval from your medical insurance company. Another important issue is that of FDA approval. The Food and Drug Administration, or FDA, is responsible for making sure that the drugs we use are safe and effective. They do the same for many medical diagnostics, ensuring proper accuracy. But what about a consumer genetics diagnostics, where they require Color Genomics to submit an application for FDA approval? Interestingly, Color Genomics has found a way around this. A doctor is involved in every order, either the woman's physician or a physician provided by the company. If doctors place orders, testing companies that operate their own laboratories do not need FDA approval to offer testing. The company only needs to provide public data on the accuracy of the test. However, there is a danger that a widely available consumer genetics diagnostic kit could lead to over-testing and potentially over-treatment. In particular, health advocates have worried that for women who don't have breast or ovarian cancer running in their families, then the benefits would be small from such a test, whereas the costs could be large. What are the costs? One potential cost has to do with the psychological stress of undergoing the test. A bigger cost is that the test may produce ambiguous results that frighten women into taking unnecessary drastic action, such as getting mastectomies or having their ovaries removed, perhaps unnecessarily. A patient receiving such a test would need genetic counseling. A woman with one of the severe BRCA1 alleles, but no cancer running in her family would face a dilemma. Perhaps she possesses other genes or environmental factors that suppress the danger from her BRCA allele. Or perhaps she is simply not aware of the cancer that runs in her family. Assignment 15A asks, what genetic diagnostic test or tests would you develop for your complex trait? Please put your answer under question 13 of your term paper. Assignment 15B asks, how will the complete genome sequence of every person change the genetic diagnostic testing for your complex trait? Put your answer under question 15 of your term paper.